Okay, also the green line disappeared, so I can start right away. So let me start with a little bit with the motivation of the study. And so the idea is very simple, so it's very general. Biological systems process information from the surrounding environment, and they do it through interconnected processes, dynamical processes that can happen at multiple spatial-temporal scales. Just to give you very general ideas of which kind of systems like belong to this very general framework, bring it into proteins. This is the case of the ABC transporters. These are transporter proteins, ATP binding cassette. So they transport roughly 10 to the 6 molecules per second, and they do it through different chemical pathways, different conformation, also guided by allostery. I'll not give you the details of this, just an idea that there are multiple timescales involved in this process, given by the rates that characterize this particular protein as many others. Yes.
Okay.
Oh. I don't know.
You can maybe just touch the screen. Sure. Maybe not.
Let me see if I can do it from, because I don't see this here. Let me.
There's cursors on the screen now. Yeah.
Okay, good. Okay, perfect. Okay, so good. Okay, this is just another example. It's an example of a bacteria that modulates the flagella motors according to the local nutrient concentration. Also, in this case, there are multiple timescales involved. The response is roughly one on the 1 second, 2 second timescale. But there are several other timescales involved in all the reaction inside the bacteria. And another example are neurons. They act collectively or they act as a single neuron. There are multiple frequencies and timescales involved. And also here there is a huge amount of information, as you can all imagine, that trigger collective responses and the emergence of a memory in neural networks. So these are just three examples of three very different systems that kind of belong to this very general framework. And of course, accurate processing is crucial to establish a robust functioning of all these systems. And this accurate processing is performed through complex operations, they can be even linear operations across these multiple timescales, and also through energy consumption. So these systems usually work out of equilibrium, they use energy to process information and to respond. These nonlinear operations and these processes that consume energy can happen in several ways. One of these is through the presence of multimolecular reactions. This is an example of a signaling network. I don't know how to actually make these things visible to anybody. I am moving further away. Okay. Through multimolecular reactions. So there are reactions involving binding and unbinding processes, catalytic reactions, and all this stuff, or through the interaction involved in ecological dynamics, or even through all the dynamics that regulate gene regulatory networks, or even in a neural network that is governed by activation functions that are typically nonlinear functions. So these are just, again, 4 examples. I'm going to discuss part of all this. This is just to say that this kind of framework is very general. It encompasses several systems in living, biological, non-biological systems. So it's a very general framework. So then there are several questions that we try to answer. And this is just a subset of those. How timescale affect information, how this nonlinear operation affect information, and how the energy consumption also is connected to information. So I'm going through all of these one by one. So let's start with how the timescales can affect information processing. Since we have multiple timescales at play, so this is the first thing that we have to understand is how this timescale structure influences how the system processes information and information propagates in the system in general. So before starting with all the results, let me give you a very brief, not really technical, but slightly more technical than all the rest of the talk, introduction to what I'm going to do. This is not really important, so if you don't want to follow the next 5 slides, it's fine. But it just gives you an idea of the framework that I'm going to use, mathematically speaking, to solve this problem. So these information processing systems can be described through a set of coupled stochastic processes. And I mainly use two different scenarios. The first one is a discrete case. In a discrete case, all these processes are n different random workers, so n different Markov jump processes. They all are coupled. And each one of these processes can explore different states. So these processes can be molecules in a chemical network or can be ecological species. Different states that all these processes can explore can be different conformational states of a protein or can be molecular counts in a chemical network. And all these processes are connected and are connected in a particular way. The way they are connected is not that I'm choosing this way because it's simple to analyze, but if you think to this kind of systems connected layer by layer or process by process, basically the idea they are connected is that the state of a certain process can influence the dynamics on another process. So this is the way two different processes can be connected in a very general terms. These are, I mean, for people that comes from the network community, these are called triadic interaction or high order interactions, whatever it is. It's not that important. It's just the fact that the state of a certain process can influence the dynamics of another process. This is a general way two different things are connected. Of course, they cannot be connected only in this way, but pairwise interaction can happen within the same process, and this kind of interaction can happen between two different processes. That's the difference. And all these processes have their own timescale. So the dynamics is a typical master equation dynamics with a certain operator. They can be even a nonlinear operator, so nonlinear in the variable. These indices will indicate the n different processes, and every process has its own timescale. So this is a very general framework, and this is the discrete case. The other scenario is a continuous one, is analogous. So also in this case, we have n degrees of freedom. They are coupled. This can be the activity of some genes, or it can be the activity of a neural population. Also, these continuous systems, they can be coupled via interactions, internal interaction between different processes. These processes can be, of course, multidimensional. And they also can be coupled through diffusion terms, like a temperature gradient that maybe fluctuates in space and influence all the processes at the same time, or can be through an environmental coupling that influence the diffusion of all this processing altogether. And also in this case, these n different degrees of freedom evolve each one on its own timescale. And the dynamics is described by a standard Langevin equation or Fokker-Planck equation. It's the equivalent description. So this is not incredibly important, but it's good to know like the mathematical frame that I'm going to use during the talk. So we have to now let's do one step back. So I'm not going to speak about these details specifically, but now let's consider these two scenarios, and we're going to ask basically how we can quantify the information shared by these different processes or by these different degrees of freedom. So they are coupled dynamically, they also share information, and we like to compute this information that they share. So, and we also want to understand how this information is generated and propagated in the system. How we do this? Through the mutual information. This is a standard quantity. It's not the only quantity, so there are several other things that we can compute. We do it with mutual information in at least for this set of things that I'm going to show you today. And the mutual information in this case is between two different workers, so different processes. And the other case is between two different degrees of freedom. So again, we have different processes, they share information. We compute the mutual information between these different processes pairwise. And the mutual information is defined this way. So it's just the Kullback-Leibler divergence between the joint probability distribution and the product of the marginals. These are the marginals distribution, it's just a definition. And basically, this is zero only when two processes are decoupled. So any kind of dependency is captured by, statistical dependency is captured by this quantity. So when this is zero only when the product of the marginals is equal to the joint. So this is a standard case in which these two processes are independent, statistically speaking. And this is the same definition for the continuous case. Okay, so this is the quantity I'm going to use for most of the talk. So I thought it was important to define it from the beginning. Okay, now let's start with the idea of timescale, how timescale affect information, and let's start with a proof of concept. So just to show you that timescale can affect information propagation, and to do this proof of concept, let me start with a very, very simple example. This is not a physical example, this is a toy model. Okay, so there are two molecules. Each of these two molecules can occupy two states, A1 and B1 for molecule 1, and A2 and B2 for molecule 2. And these two molecules are coupled through this coupling C, which is, in this case, a catalytic coupling, in the sense that when molecule 1 is in the state B1, it can accelerate the transition from A2 to B2 in a catalytic way. So it just accelerates the rates, okay? It's a very simple example. It's not gonna necessarily reconstruct any physical process that I have in mind, just two entities that interact in this way through this catalytic coupling. And in this case, what we can do, we can compute the mutual information between these two processes, these two layers, if you want to see this as a network. So process 1 and process 2, okay? So the mutual information between these two processes is defined this way, as before, and capture the information that these two processes share. So they are causally coupled, so there is a coupling between the two. So they, in principle, I mean, intuitively speaking, they will share some information, right? So I mean, there is a coupling between the two processes. But we can study the mutual information as a function of their relative timescale, because we are interested in the timescale of these two processes. Timescale of process 1 is tau 1, process 2 is tau 2. So we can study this quantity as a function of tau 1 over tau 2, so the relative timescale. What we see is that there are two limiting scenarios, and the first one is the case in which the process 1 is faster than process 2. And the other one is the case in which process 2 is faster than process 1. So in this case, what we see, okay, let's start from the other case, the simpler one. When process 1 is lower than process 2, what you can see is that there is a non-zero mutual information, right? So it's what you expect, there is a coupling, and the mutual information between these two is non-zero. But when process 1 is faster than process 2, what we see is that even if the two are coupled, the mutual information is actually zero. And it's zero because since process 1 is faster and in this limit is, let's say, infinitely faster or much faster than process 2, this second process only fills the average distribution of this quantity. So it cannot really take information on this process, even if they are coupled. So they are, in fact, statistically independent. So they are causally dependent, of course, because they are coupled. The dynamics will be different from the case in which there is no C, of course. But at a statistical level, in the sense of the distribution, these two systems are decoupled. So the mutual information is zero. Okay, more or less, I mean, I think this convey the message that timescales are important, yes?
I think of this as sort of like a mean field, because the switching in process 1 goes so fast, then process 2 just sees the mean field of process 1.
Yes, yes, exactly. It's exactly what happens in this case. So in the end, the way you solve this, mathematically speaking, is through timescale separation. And in the end, you just reach a steady state for every given value of the other process. So in the end, you just see the stationary distribution of the other. So that's the reason why they are effectively decoupled. So Then the bottleneck of all this is that the bottom line of all this is that the relative timescale of two physically separated process affect the mutual the information shared between different layers. Okay, so we understood that actually timescales are important to understand information propagation in a multi-layer system. And so I want to now extend this framework to the general case in which there are not just two layers, but multiple layers are coupled together and different layers. And this is just a pictorial representation of n different processes coupled together. These are all the possible couplings that we can have in the system. For now, you can just forget about all these colors. Basically, the red ones, if you just in a nutshell, the red ones are those that goes from fast to slow, and the blue ones are those that goes from slow to fast. Okay, but it's just they are just coupling. They are all the same. Physically speaking, they are identical. We just order them from fast to slow, just pictorially speaking, and this can be represented this way. So it is a very compact representation of these n different processes coupled together through a different causal structure. Indeed, the causal structure is determined only by these direct feedback links. These are just names for these two kind of links that we have in the system. Direct are from fast to slow, and feedback from slow to fast. This is just name we decided to assign to these two different kinds. Yes.
The slowest is connected to like the one before slow, is just that the slowest can turn to the fastest, or it's just hierarchy by that you go from a slow number one to a slow number two, or fast number one.
Can you?
So if you have, for example, tau one,
Yeah.
time scales from tau one to tau n.
Yes.
Is it that the degree of freedom associated with tau one is connected to tau n, or is just tau one is connected to tau two and then tau two to tau n?
No, no, no. There's no. I mean, the hierarchy is only in the time scale, but the couplings can be whatever. So if you have n different processes coupled whatever you want, you just order them in a certain way, and you just draw the coupling that are there. So indeed, in this case, n can be connected to two, but I can also have the one connected to n and n to whatever, and one and two are disconnected. It's just a way to order the system graphically speaking. So it's not connected to the causal structure. You have these n processes, you order them, and the causal structure is the coupling, basically. And the functional structure, what we name functional structure, is determined by the mutual information shared by all these pairs of processes. So 1 and 2 can share some information, 2 and 3 can share some information, 3 and n can share some information, and the information that they share is the functional structure. And the causal one is only given by the coupling. So these two things are not necessarily connected in the sense that, as you saw before, you can have a causal coupling and no information, or you can have no causal coupling and information, like through all the other connection. So these two things are not necessarily are connected, but they are not the same. Okay. So we have the evolution of the system, exactly the same one that I showed you before. So we have the evolution of the probability, the joint probability distribution of these n degrees of freedom coupled together. They evolve at the same time to an operator which can be nonlinear, nonlinear in the variables that determine these processes, of course. And the question is, can we say something on the probabilistic structure of this system and then on the information propagation in this system without knowing anything on the dynamics? Of course, the answer is yes. Otherwise, I wouldn't be here to tell you this story. So yes, we can say something. And in particular, we exploit the presence of multiple timescale to perform a timescale separation approach and determine a probabilistic structure of the system. So to show you how this is done, I'm not going to into details of timescale separation. If you want, I can go into details later. But let me show you directly the results of all this procedure. And let me show you the results in two different, very simple examples, but they are kind of representative of what's happening in the system. So consider three timescales. Okay, these are just the basic case in which you can see some interesting phenomena. So 1, 2, and 3, they are coupled together in this way. So 3 to connected to 1 and 2 to 3. 2 and 1, 1 and 2 are disconnected. So to go back to the question, the previous question, there's no connection between the fact that they are ordered and there is a causal connection between the two. So what is the probabilistic structure of this system? It's actually very easy to find. So through this timescale separation approach, which is anyway very easy to perform, what you see is that the joint probability distribution of this system takes this form. So you go from the fastest timescales, fastest timescale, 1, you solve 1 given a given value at a given value of process 2 and process 3. That's why this, exactly as before, basically 2 and 3, they both see 1 only at an average level. They see the entire distribution of process 1, right? So process 1 reach a steady state, while 2 and 3 are fixed. So the steady state distribution of 1 will be conditioned on the value of 2 and 3, or a value specifically on the state of process 2 and the state of process 3. So in this case, 3 influences 1. So the steady state of process 1 has to be conditioned on the state of process 3. So the structure is this one. So 1 given 3. Okay, so for any value of the process, of the state of process 3, we have a different distribution for process 1. So we can only define a conditional structure. We cannot, of course, solve for PST1 independently of the rest of the system, because there is this connection. Then we pass to the second fastest, 2. But 2 is independent of anything, right? So 2 does not depend on any other slow time scale. Does not depend on 1. And 2 influences 3, but not the other way around. So we can actually solve 2 and find the steady state of 2 without any conditional distribution. So we have just 2, P of 2, stationary state. And then we pass to 3. What happens to 3? 3 also is independent, but there is 2 that is influencing 3. This has another effect, which is basically the fact that P3 is not following autonomous dynamics, but is following a dynamics that is influenced by the steady state distribution of 2, which is fastest. So since 3 is actually perceiving the steady state distribution of 2, is feeling the whole distribution of this fastest layer, the operator that governs the evolution of 3 is a different operator. Of course, I mean, it's, we don't know the operators, we should know the entire dynamics, but we can write some effective there. So this just means that the operator that influences the dynamics of 3 is not the operator of 3 alone, but is the operator of 3 alone integrated over the distribution of 2 that is influencing 3. Okay, that's all. It's actually very simple. So feedback interaction gives you a conditional structure. Direct interaction will give you an effective operator. That's in a nutshell. From this distribution, we can actually compute this quantity here, which is the mutual information matrix for these multiscale observables. I don't know if there are any Italians in the audience, but usually this is a very funny joke, because Mimmo is an Italian nickname, which is a very famous one, but it's also the name of this matrix. So we compute this MIMO, mutual information matrix for multiscale observables, which is the matrix that captures which information between any pairs of states is non-zero. In this case, only the information between 3 and 1 is non-zero. Since if you take the structure and you compute the mutual information, you can see immediately, then the only mutual information that survives is the one from 1 and 3. All the others go to 0. Yeah, exactly. Yes.
Typically, the composition remind me about this BD propagation or message passing technique in static, you may know it very well. And then the only limitation of that general decomposition is because is that if you have a cycle network, it breaks down. So in these two cases that you demonstrate, is the acyclic graph, right? So what happens if you have cycle then?
Cycles, you mean maybe 2 to 3 and 3 to 2?
And 1 back to 2, for instance.
In principle, there's no problem. You just have to remember that every direct connection gives you an effective operator, every feedback connection gives you a conditional structure. If you remember this, you can perform all the calculation even in presence of a loop. There's no problem. I mean, of course, it might be more complicated, and this will be probably a full matrix. since when you have a loop, everything is connected. Basically, you connect everything with this loopy structure, but you can do the procedure. So we did it so for several loopy systems. Yes.
Assuming that the faster timescales are always much faster, or
Timescale separation means exactly that. So infinitely faster, specifically infinitely faster. So each one is infinitely faster than all the others. So they are all infinitely faster than the other one. So it seems like a very strange assumption, but in the end, it's not that wild. So if you do a simulation, one usually we check that one order of magnitude for for reasonable systems is usually okay. There is also some other paper that use similar techniques for chemical systems, and they also claim that one order of magnitude is usually okay to have a good approximation. The point is that when you do, imagine you do this for time series, for example, right? Now, you have the statistics of the states and you want to compute the mutual information, you will never get zero. Even if it's really zero exactly, you will never get zero. You will get a very small number. But what we saw is that the structure of this matrix is actually preserved. Even timescale separation is not exactly in this infinite limit. So things that are big in the limit of timescale separation stays bigger than the others when timescale separation is not exact, of course. Unless you arrive to the case in which all timescales are the same, and in this case, of course, you cannot say anything. But as long as you have some separation of timescale of one order of magnitude more or less, you still can see that some structure emerge, and you never get zero anyway when you do this from numerically speaking. So now let's, okay, so basically we can already draw two conclusions from this very simple example. The first one is that feedback interaction, this kind of interaction, so from slow to fast, they generate information in the system, while direct interaction alone, they do not generate information. Indeed, information is only between 3 and 1. Is the two states connected by this feedback interaction, not between 2 and 3. Then what is the role of this direct connection? We can see in these other examples, just move just one link from here to here, from 2 to 3 to 1 to 2. We move just one link, one direct connection. The structure completely changes now. You can follow the same procedure as before to derive now the structure is this one of the probability. And from this structure alone, you can already see that the model information in principle is non-zero everywhere. Of course, this value that we draw are for a specific dynamics, of course. So the specific value that the information attain depends on the specific dynamical process, but the fact that it's zero or non-zero only depends on the causal structure and on the time scale ordering of the network. Okay, so this actually highlights what is the role of direct connection. Direct connection can propagate the information that has been generated somewhere in the network. So you generate some information from slow to fast, and then you can propagate this information to all the fastest layer through direct connections. Okay? So this is the role of direct link. So feedback generates something, and direct link propagate this information. Yes?
So where did they go? Metric. So how did you draw the arrows on the?
This arrow.
Yeah.
This arrow is basically emerging from this from the time scale ordering. So it's not really model information is a symmetric quantity. So there's no really directionality in the information itself. It's only in the ordering of timescales. So this is it just follows the interpretation that this is generated from 3 to 1 and then propagated downstream from 1 to 2. But this is it's following the timescales. It's not really connected to the mutual information.
Okay.
Yeah.
Is it the way you're doing the timescale separation, why you only get pairs? So that you never have these triplet interactions?
We can have those. If you can have a more complex conditional structure here. We only have pairs because of these very simple examples, but we can have something much more complicated. But the mutual information, the reason why I choose this specific example with just pair interactions, the mutual information anyway is between two processes. So this is the minimal case in which you can see these three rules emerging when you have pair interaction. Of course, if you add more stuff, it's much more complicated, and you still see the three rules, but it's just slightly more difficult to pinpoint those from more complex scenarios. But I mean, yes, in principle, you can have if you have more feedbacks going to one, for example, from three, from two, from four to five, you can have all these states here appearing as conditional in the conditional structure of the probability. So you can have something more complex. Okay. So I hope I convinced you enough that timescale are important to modify propagation of information in these kind of systems. And I think we can actually move to the second part. So what we can learn from this timescale structure? Can we say something about the functional structure of the system? In general, yes, so we can say something. So if you have a specific model in mind, now I'm going to show you two specific examples. One will be very pedagogical, not necessarily very insightful. The other one will be actually more interesting, at least to me, on what we can learn on this kind of system. So I'll go fast on the first one. And the first one is just of a very simple structure of a chemical signaling network. So generally speaking, they are multi-component units that takes a signal and convert it to a production of specific molecules. This is a very general definition. I'm not going to the details of describing any specific signaling network. This is a very simple structure of a signaling network that I designed with a receptor, a certain readout unit, this is the production of this specific molecule, and a certain unit S which store information. This kind of structure emerged spontaneously in several papers. I also try to find what is the best architecture to have habituation dynamics, adaptation dynamics, and any other observed dynamics in this kind of signaling networks. They always this three unit structure emerge with there always a receptor, of course. There's always a readout molecules. It's the one that you actually read in the system, and there's always another unit that has the role of storing some information in the system. Okay, so what we can say about this kind of system, then of course there are some connection that we can put between these units, and these units can be connected in several ways. There are cases in which the receptor influences the readout activates the storage, and the storage inhibits the readout, or the structure can be slightly different. So the input arrives to the receptor activates the readout, but the receptor also activates the storage, and the storage inhibits back the receptor. So these are two different causal structures in the network. We chose a Bertrand dynamics to simulate this system, just a choice. It doesn't really change anything in the qualitative picture of the system. And the results are these two. This is the dynamics of the system, where these gray bands are where the input arrives. This is just to show that if you only see the readout, the system is exhibiting what is called habituation dynamics, at least in this very simple scheme. So when you have a repeated stimulus, which is identical, you see that the activity of the readout decreases in time, and then eventually can spontaneously go up again after a certain time. So from these two schemes, only from the readout, the two systems respond similarly to this kind of input. But then we can analyze these. These are already ordered in the time scale from fast to slow as before. So we can analyze the functional structure emerging from this system, and the functional structure is actually very different. So in one case, you have that receptor and storage, they are not connected functionally to each other. So the distribution of receptor and storage are completely independent. Even if the dynamics are connected, they're connected to the readout. While in the second case, storage and readout are independent, probabilistically speaking, but still there is some information that passes from S to U through the readout process. So there may be something we can learn on the functional structure of the system at a very general level by using this kind of framework. This is a very pedagogical example just to show that this can be applied to, for example, signaling network. This is one of very simple. We did a much more detailed system that I'm not going to show you today. of a signaling network. But let me pass to the second example. It might be more interesting to show you what we can learn. Yes.
The second one, the B case is called negative feedback, right?
This one?
No, the B, the second plot, the second motive, sorry.
Here,
It's,
yeah.
It's the negative feedback loop, right?
I never remember the names of all these things,
Because
but might be,
it's my-
might be. No, these two structure have like very specific names, but I never remember them. So sorry, but might be that one of them is negative feedback loop. Yes. So these are two structures that both of them works to exhibit habituation. So yes, I mean, it might be that this is the one that is called negative feedback loop, but I have to be honest, I don't remember the standard names for these internal structures. All these names have been proposed, I think, in one of these papers that I showed you before. Yeah, exactly. So they propose some of these names of this structure. Yeah.
If to have a robustness system in this signaling pathway, so one is a negative feedback, I think is the second one.
Yeah.
And the upper one is that so-called incoherent feed forward.
Yeah, not all the structure that they propose actually exhibit habituation. So this is a tricky thing. So they propose something that exhibit robust adaptation. This is a different story than all this, but not all the structure exhibit actually habituation dynamics. So I choose just two of these examples that exhibit habituation dynamics. That's why I usually do not stick with this definition, because there are some of the network that they highlighted that exhibit robust adaptation, but no habituation. So that's why I don't usually stick to this kind of definition, and I prefer to like just draw the network. Okay. The second example is about neural population dynamics. This I think is slightly more interesting, at least to me, in terms of modelization also, because I mean, the things that we found are kind of connected to several experimental observation. I'm not going to discuss in detail today, but let me go over the example, this specific example. So in this case, we model the system as a two population system, so excitatory and inhibitory populations. These two are connected and are connected through these internal interactions. They may be linear in the very simple case, so there may be non-linear interactions, usually modeled with a hyperbolic tangent. And there is also an external input, h of t, and we take this input as a switching signal. So it's a signal that in this case it just switches between two states, but we can make this switch within n different values of the input, and the input can switch with its proper timescales, while the internal population evolves on a different timescale. So we have two different timescales: the timescale of the input and the timescale of the internal population dynamics. They are coupled through these inhibitory and exhibitory interactions. And what we can study is the modal information between the population X as a vector, like E and Xi, excitatory and inhibitory, and the external input. So the modal information that the input is sharing with the neural activity in the system. And also in this case, of course, we have that when the input is very fast, the neural activity it's basically seeing the input is seeing an average input. So it cannot really store any information on the input statistics. It can only see its average. So it's just one value. While in this other case, when the neural population is actually faster than the input, in this case, there is information that the neural population can store on the input statistics. Since now they are seeing the whole statistic of the input, which is very slow, and not just an average level. And so, as before, the modal information is non-zero when in the limit where the input is lower than the population, and is zero when the input evolves much faster than the neural population involved. Then we can study in the parameter space of the system, we can study what happens to this information. This is just the linear case, in the case in which f is a linear function. So, and in this case, as you might imagine, what happens to this mutual information is that it basically picks where it actually diverges when the system is at the edge of instability. And this is, mathematically speaking, this is very simple to understand. So there is a network is unstable, so every response is actually an exploding response to small modification in the system parameters, and this also happen. This is reflected in the mutual information. So that when you have a change in the input, you can see the system responds infinitely strong to every change in the input. So the information also explodes at the edge of instability. We can actually cure this kind of explosion by adding an hyperbolic tangent. When the system is nonlinear, the peak still remains, but what happens is that the system, of course, is not unstable there at this point, and you can actually go in the region of the linear instability, and you see that the information goes down again. And it goes down since the system is actually reaching this saturation regime of the hyperbolic tangent, and then the information goes down again. So we can actually learn something on this information encoding on this near population given an input. This is the limit in which the input is slow, so that the information is actually non-zero. And we can see that there is some interesting phenomena that are appearing at the edge of linear instability in this system. Then we can actually go a little bit further in this kind of example, and we can add another layer, another timescale, and restore the case of a 3 timescale scenario, which is one of the more interesting case in this to study multiscale information propagation. And the other timescale we can add is the plasticity timescale. So plasticity is what modulates the strength of this interaction, and in this particular case, we chose plasticity to modulate the excitation strength of the interaction, which is this W here, while K is the parameter that controls the fraction of inhibition in the system. So this is the way we encoded plasticity in the network, and plasticity follows its own dynamics. This is a simple Hebbian, what is called Hebbian learning rule for the plasticity. We also tried other learning rule. Nothing really changes qualitatively, so I'm going to show just the Hebbian one. So now we have 3 timescales at play. We have the input, which is still a switching input. We have the timescale of the neural activity, and we have the timescale of the plasticity. So as I said before, the input has to be slow, so that the input can actually propagate information in the system, but we still have some variability in how we pick the timescale of the plasticity. So we can have a slow input slower than the neural activity, and a plasticity which is even slower than the input. This is one case. The other case in which is the case in which these two are switched. So the input is lower than the activity, so the input can actually propagate information in the neural activity, and the plasticity is faster. So let's start with this case, the case in which plasticity is low, is slower than the input at least. So a slow plasticity integrates the entire statistic of the input. So now the population can actually see the input that varies, since it is faster. So the population can encode information on the input, but plasticity only see the whole statistic of the input, since it's even slower. And in this case, since plasticity is defined by the entire statistic of the input, we can find a value of P, which is the parameter that governs the dynamics of the plasticity, that leads to a stable value of gamma, which is indeed the plasticity modulation value, the plasticity strength, that gives also to a modification in the interactions, and then leads to a certain value of the mutual information. And what we can find in this scenario is that we actually, since the plasticity uniquely determines a modification in the strength of this coupling, given a statistic, a whole statistic of the input, we can actually study what is the value of the plasticity that maximizes this information. Since this just depends on the whole statistic of the input, we don't have we can do this kind of maximization even if the input is a switching undergoes a switching dynamics. And in this case, what we can see, I'm not going to details of this scenario. I just want to show that it's a rich phase diagram in this case, since this plasticity dynamics also induce multi-stability in the network. So the dynamics, the phase space is very rich, But what is very interesting is that if we pass to this slide, probably this is better to explain the idea, is that in this rich phase diagram, we can have that plasticity, an Hebbian plasticity or an anti-Hebbian plasticity, they both can be optimal to reach maximal information. So in some part of the region, the system has to do some reinforcement on the internal coupling. In some other part of this phase diagram system, actually to reduce the strength of the coupling to learn more on the external input. And this is just given by the fact that the plasticity is lower, and this can actually perform a global control on the entire network. How to actually modulate this experimentally, not entirely true, but in principle, there is some studies on the fact that there are modulation of the some neural, I think they are called neural modulator specifically. They can actually, it's believed that they tune this plasticity strength in the network. So, yeah.
So this is the case for tangent hyperbolic ring.
This is for linear case. For tangent, for the nonlinear case, does not change much the picture. The only difference appears in this region here. I mean, that's basically the two things are the same in terms of information, as long as you don't hit the saturation regime of the hyperbolic tangent. And in this case, you see some modifications. So in the hyperbolic tangent case, we didn't do the study very carefully, I have to say. But what should happen is that in this case, you should also see some anti-Hebbian plasticity as optimal behavior, as optimal strategy when you have the hyperbolic tangent.
In the class paper, you see also different regime where you can have different type of chaos. And you also see here.
Here that we are studying everything at steady state, and the dynamics is also stochastic, intrinsically stochastic, so there is a noise. So there is no such thing like, and also there are two populations. So just two populations, coarse grain population. So it's not the same dynamical picture than is in the Clark scenario. So we're not studying really the dynamics of a multi-population system, and also we are not studying the case of random interaction. So here it's just a two-model system with just two parameters, just global parameters. This is a very classical modulation for people doing experiments in neural systems. So with just two population or two or more than two, but coarse-grained population. So it's not like completely wild approximation to use. Let me go quickly over also to the other case, since I want to show that actually this idea of timescales might have an impact on what we understand on the system. And this is the case in which plasticity is faster than the input. So in this case, plasticity, of course, plasticity modulation depend on the instantaneous value of the input. So now plasticity is faster. So it will not integrate the whole statistic of the input, so it cannot drive the system towards a global optimal point. It should be an optimal point for every single value of the input, and of course it's not a global optimization. But what happens in this case is that this multi-stability, which is induced by the plasticity dynamics, can actually be exploited to store some information and to perform sequence discrimination. At least you can discriminate the ordering of the sequence spontaneously. What does it mean? This is just one example in which we picked three different values of the input. So this is remember this is a switching input. So now it is switching between three different values. These three. And these are just three different values of the plasticity parameter P. And the region that you see in gray is the multi-stability region. So what you can say basically, what you can see basically is that when imagine that you start from this intermediate sequence, imagine that you start from the lower input values, and then the system which to the multi-stable regime, and then the system hit the lower value in this multi-stable, in this bistable regime, then is switched to the higher case, the system jumps to a higher value of the plasticity of gamma, and then you go back to the multi-stability case, for example, and then you end up in the upper attractor of this bistable regime. But if, of course, if I present the system a different sequence, maybe first the upper value and then the lower value, and I start from the top instead of from the bottom, what I see is that the system first hits the upper value of this bistable regime, and then it goes down, and then it's the lower attractor of this bistable regime. So bistability helps in discriminating the sequence in which I'm proposing these different inputs to the system, and I don't have to perform any tuning here. So I just have to put the system in the right spot so that multi-stability can be exploited without any specific tuning to optimize the, let's say, the learning of the discrimination, as I don't know if you want to call it learning. And this is the case, just the simple case of two different sequences at the top of these small panels. There are two sequences that they correspond to different sets of plasticity that are explored in the network, and also to different responses in the neural activity. We can quantify everything through mutual information. In this case, between the neural activity and the whole statistics of the sequence that we are proposing to the system. And this is, of course, non-zero in a specific region. And this region is the one in which multi-stability can be actually exploited to store some information on this temporal ordering of the sequences. And again, this is solely given by the fact that the plasticity is at intermediate time scale. Okay, so we also answer to this question. Actually, I don't know if I can answer to all these questions in this talk, but let's try to go on to start to discuss a little bit the effect of nonlinearities. So already in this case, we saw that nonlinearities have some effect in like reducing the information when the system hits saturation regime. So there are some non-trivial effects due to the fact that the system can have a nonlinear dynamics. So in general, linear processes are already capable to do a lot of stuff in terms of information. So if I couple the processes, lunge event dynamics, lunge event like the n degrees of freedom that follow each one lunge event dynamics, if I add a linear coupling, I can already do a lot of stuff. The system can encode the environmental information, can transduce information from hidden variables, and they can perform a lot of things already with linear dynamics. Then, if you add a pseudo-linear dynamics, you add a little bit of like a catalytic reactions. You can also see that the system can perform some classification. This is a recent paper that just appeared, not mine. But it's very nice, and it shows that actually a linear system with some catalytic reaction can actually perform some classification. But if you also add nonlinearities and we also add the presence of fast reaction, you can actually show that the system can really approximate any dynamics. It's just a much more complicated task than just classify a certain input. So somehow this is just to show that nonlinear dynamics can actually enable the system to do much more than just classification or just encoding some information. So with this idea, let's study this effect of nonlinearities again in a very general framework. So again, we have 3 layers that you might have understood that this is a basic framework that we're going to use for study these kind of systems, because it's a minimal framework in which you have some interesting phenomena that appears in terms of timescales. And we have an input, a processing, and an output layer, so very general. And the processing happens through timescales. So this is the fastest, this is the slowest. Actually, then we're gonna see how these things are organized, but these happen on 3 different timescales in general, and all these processes are coupled. So imagine that you have a signal, a multidimensional input signal that is transmitted to the processing layer through a certain transfer function, and then signal is transmitted further to the output layer through another transfer function. So you have these transfer functions that are nonlinear in general, and we consider also the coupling inside the layer to be linear, just for simplicity. So this is the whole dynamics of the system. So you have linear intralayer operators. You have nonlinear interlayer transfer operations in this very general form, and then you have the noise for each layer, and each layer has its own timescale. So timescale of the input, timescale of the processes, timescale of the output. Okay. Also in this case, we quantified the mutual information between the input and the output in this form. So this is just, again, the Kullback-Leibler divergence between the joint and the marginals, same definition as before. And we want to basically understand how much the mutual information between the input and the output is controlled, is influenced by this nonlinear operation through the processing layer. So again, the input has to be the slowest variable at play, since we want to have some information generated by the input. So as always, in this case, the input has to be a slow variable. But even if the input is slow, we still have some freedom to choose the processing, as before the plasticity. Also in this case, we can have the processing which is faster than the output, or a processing which is slower than the output. And in this case, we can have, then it's faster, this is slower. Sorry, I mispointed. We can actually study these two cases, and we see that they do lead to different results. But we also have another variability on how we pick this transfer function. And in particular, we actually choose between two different schemes, which are particularly popular, sometimes even used interchangeably to some extent. One scheme is nonlinear summation. Nonlinear summation means that you have all this set of inputs, and all these inputs are nonlinearly transformed, each one, and then they are passed to the other layer as a summation. So you sum all these nonlinear transformation, and the other scheme is a nonlinear integration. So you take all these inputs, you sum them all together, you create a unique input, and you pass this input to a nonlinear function, and then you pass this to the other layer. So these are two very different schemes, even if they are kind of used interchangeably, and especially when you do some linear expansion, that these two lead to very different results, very different efficiencies, at least in terms of information processing. So let me do one step back, and let me start with the case without processing. So we have just an input and an output, that's all. And in this case, we can compute the performance of nonlinear summation and nonlinear integration. And to answer the question that one you asked before about the Clark paper, in this case, we actually have a large network, and we have some random coupling between the layers and inside the layers. So we pick the intralayer interactions from a Gaussian distribution with zero mean and certain variance, and the interlayer interaction from another Gaussian distribution with zero mean and another variance that controls the heterogeneity of these interactions. So here we have some random part in the network, and we do statistics over all these interactions. So we don't use the MFT tools here. We just do numerical statistics for every set of these values, and then we average to obtain a value of the information at a certain given value of the other parameters. So we studied the performance of summation and integration, and we compared the two in terms of the strength of coupling and heterogeneity between input and output. And what we see is that there is a crossing behavior from the case in which summation performs better to the case in which integration actually performs better in encoding information. At this level, this just means that you have a certain set of parameters with a low heterogeneity and for almost independently on the coupling, in which you have that integration performs better, and you have another regime for large heterogeneity of the coupling in which summation performs better. Now, let me add the processing into the game, and let me add the processing as a one-dimensional processing. I want to do minimal change to the system to make some modification, and I add a processing which is one-dimensional. In this case, basically, I move all the parameters from the input to the output, to the input to the processing, and then I study the system as a function of the heterogeneity and the coupling between the processing and the output, which is basically the pieces that I just added to the system. When we add these pieces, we add the processing into the game, what we see is that there is a huge difference between the fast and the slow processing case. So the slow processing case seems to be always not really performing well with respect to the no processing scenario. So if the processing is slower than the output, it's actually better to have no processing at all, and at least in terms of mutual information. And while if the processing is faster, you can actually, there is a threshold of coupling in which you can actually outperform the case without processing. So a fast processing enables higher mutual information with respect to the no processing case. Consider that having a processing is not just the same as condensing everything into a unique layer and considering an input and a larger output. So processing has its own time scale and has its own its own dynamics and its own transfer function, its own coupling. So it's a much more complicated than just including things in the output or in the input. So it's a completely different thing. So we're gonna focus on the fast processing case, which is actually is the more interesting one between the two. And in this case, we can actually go to larger systems, not just smaller ones, the one that I show for one-dimensional processing. And what we see, it seems that for when the system is large, when the processing is large, it seems that integration is generally favored in the sense that integration performs much better than summation. So the two different linear schemes are actually very different in the sense that for large system, nonlinear integration seems a much better scheme to process information. Of course, heterogeneity plays a big role. So if you increase heterogeneity, you can still have a certain switching behavior. And interestingly, also the dimensionality plays a very big role. So if you increase the dimensionality, you push the system towards better performance with integration. And in particular, this can actually be seen in a more proper way, studying the role of the system dimensionality. This is just a slide to see that I'm going to do this. Nice. So if I increase the processing dimensionality, what I see is that the integration performs better and better as I increase the processing dimension. So it seems that integration is actually favored for large processing systems, while summation might be favored for in the small processing case. So it's actually these two different schemes can be optimal in two very different conditions for large and small processing systems, which are actually two very different classes of systems that one can study in this kind of information processing scenario if we think to the entire set of systems that actually follow in this category. We can also find an optimal dimension. This is also another interesting stuff. So changing the dimensionality of the input, the dimensionality of the processing at which the information is maximized also changes. And in particular, we see that larger input are typically associated with smaller processing and smaller inputs to larger processing. This is at this level, is just an interesting observation, but this is an observation that reminds a lot of the two like competing schemes, which are the high dimensional embedding. So you have something very big something small you want to embed it in a high dimensional space, or the other scheme in which you basically do low dimensional projections. So you have a large input, you project it into a smaller processing. So again, these two schemes seems to emerge kind of spontaneously as two competing strategies when you want to perform efficient processing, at least at the level of mutual information. So with this, I concluded the part on the effect of nonlinear processing. So nonlinear processes, nonlinear operations can actually changes when they are coupled to a certain timescale ordering. They can change the processing operation of a system. And I also think that I'm out of time. I should be.
10 more minutes.
Okay, 10 minutes. I think maybe they can be enough. So I can also sketch a little bit this other part, which is about energy budget. I'm going to be quick. So this is a slightly different scenario that to understand a little bit what is the role of the energy budget in the system. And this is the scenario basically. So it's a sketch, of course, but imagine that you are on this side of the door. So you're listening to some music. There's some other guy playing a guitar on the other side of the door. The door is shut, so you cannot see the guy. And if I ask you, okay, what is the age of this guy? What is the nationality of this guy, or any other like features of the guys playing the guitar? It's not really easy to answer, right? So maybe you hear some, I don't know, some old Italian songs. Okay, maybe the guy is Italian, maybe he's old, but we cannot rely on a model. We have limited information to really reliably answer to this question. But we can actually map this scheme into a very simple biological model, very simple one. I'm going to give you at the very end, an example of a real biological system that fits into this scheme. But for now, let's keep this general. So this guy is playing the guitar, can be an active bot, some activity, internal activity in a membrane. The music is the membrane, so is what is transducing the information from the inside to the outside. And we, or the guy who's listening to the music, is an external chemical probe, an artificial probe, whatever it is. It's something that is only feeling the membrane without being directly coupled to the active bot inside. So this is the kind of scheme. And in this scheme, we have this set of couplings, so bot to membrane to probe, and we want to transduce the information from the target, which is the active bot, to the probe. So we, or the probe, would like to get information on the active bot without being directly coupled to the active bot. Okay, this is the goal. How we can do this? Imagine to change the properties of this probe. The only thing that we can change, this is a system which is, everything is linear here. So we have only linear coupling. We did also an extension with non-linear coupling, nothing changed so much in the quantitative level. So let me stick to the linear case, which is much simpler to read and understand. We can change this parameter of coupling between the probe and the membrane. And we can choose this parameter to be such that the information between the probe and the bot is maximized, while the entropy production that is induced by the fact that we have a probe is minimized. This is an assumption. So there's no biological indication that says that this should be the right function to minimize, of course. This is just an assumption. Basically, the target is that we want to maximize information. We need to put something that counterbalance information. Otherwise, if you just maximize information, this coupling explodes. I mean, the information can go up to infinity. So this coupling go up to infinity, and we have no physical system. So we have something that balances this coupling, this value of this coupling. If you say just put some quadratic function, you see that the solution that you get as a diverging dissipation. So then we decided to put explicitly some entropy production there. So something quantifies the dissipation into the environment. It's a proposal, not a can be eventually tested, but I mean it's a proposal at this stage. Lambda here represents the strategy of the system and also sets the energy budget that the system has to perform this transduction. We have also in this case 3 different time scales, and also in this case the active bot has to be slow, since the active bot has to put information in the system, same as before. Well, in this case I can maximize this functional, and I can obtain a solution for an optimal coupling, and I obtain a certain solution for the information and the entropy production as a function of the sigma, which is the parameter that controls the coupling between the active bot and the membrane, which is the only parameter that we still have at play since we maximize over A, the other parameter. Okay, nice. So far we didn't learn so much on the system. We just learned that transduction is possible. But actually I cheated. I say that the system is not actually doesn't know anything about the membrane, sorry, about the active butt. And still, the system is maximizing a function that explicitly takes information on the active butt. This is the mutual information between the probe and the butt, and this is a total entry production of the system that, of course, keeps track of the active butt. So I'm actually using an information that I don't know to maximize and to set the probe to the right coupling. So what I should do, or if I imagine to be a certain probe or a biological probe that has to take information on the available things that the probe can observe, I cannot use this functional. I should use this functional instead, this other one. And this functional is very different from the previous one. It only uses the partial information available. So it only uses the mutual information between the probe and the membrane, which is the only thing that is available to the probe. And it only uses the entry production estimated from the trajectories of the membrane and the probe. This is the only available information. We can analytically estimate these two quantities. Fine. I'm going to skip over how we do this, but there is an analytical estimation for both of them. And we can build a new functional, which is this partial information functional, and we can actually maximize this one to see, is the transduction really possible using partial information, and to what extent? Well, the solution is, this is the previous solution, and this is the new one. So transduction is actually possible, and actually, in terms of information, transduction with partial information, sorry for the repetition, is actually higher than the transduction with an all-knowing probe. This might be might seem like a very counterintuitive case, like it be a paradox. I'm maximizing something which is not my target, and still my target function is higher. Why? And the reason is that we are actually losing in dissipating more. We are dissipating much more than the previous case, but this dissipation is the best we can do. We cannot reduce dissipation much more than that, since we don't have access to the information to the dissipation between the butt and the membrane. So we cannot control this part of dissipation. So we can reduce dissipation as much as this value, and still we can get an information between the probe and the active butt, which is higher than the ideal case, at least in some regimes. Of course, this can hold only in certain particular regimes. At some point, the system hits the case in which there is no efficient transduction anymore. There is an inefficient regime in which this curve is lower than the ideal case. And then there's also a case of no transduction. Why these two cases happen? And the reason why they appear is that because the entry production, estimated from the partial trajectories, is much higher than the real one. And so when you try to minimize something which is much higher, the system just say, okay, I'm dissipating too much. It's better to just get zero information on the system. So my optimal point is actually getting no information at all. So this effect of having partial information kicks in at some regimes, but there is a whole set of parameters in which this partial information is actually better than knowing everything, at least in terms of the information we can get from the system. We can do some, also in this case, we can do some exploration depending on timescales, and we can see that as we change the timescale, we can have these regimes of inefficient transduction and non-transduction and change a little bit. And there are some cases in which basically we have no efficient transduction at all. We can only hit the case of inefficient and non-transduction. So actually the phase space in all parameters is very rich, but I'm not going to the details of this part. And this is just the case that I finished, just 2 slides. This is the specific biological case that falls in this category of 3 Langevin processes coupled, in this case, also in this case linearly coupled. This is a red blood cell. This model has been proposed very recently in this paper, and they show that this model captures the entry production of this red blood cell in the proper way. If you do not agree, do not complain with me, not the author of this paper. Um, but I took basically the data that they have and also the parameter that they estimated and they fit to match the entropy production. So take this model and consider the fact that basically they measure only external fluctuation, external membrane fluctuation, what is called the flickering of the external membrane. And this flickering is believed to reflect the activity of the internal cytoskeleton, basically. So this is again a transduction process. So you have something that you don't observe, and you have something that you are observing, and you want to understand how this transduction operation works with respect to an ideal case in which you know possibly everything. In this case, you know the entropy production, so you actually want to solve the inverse problem. You know all the parameters. You want to solve the problem is that given the parameters that I know, what is the strategy that the system is implementing to perform transduction as I am observing. Putting all these things, all this framework with the data all together, what we can see is that there is a set of cells that are passive, so they produce less entropy, and also the information is very low. And there is an entire set of cells where the information is higher and also the dissipation is higher. And in this case, we see a very interesting observation if we compare the transduction scheme with the ideal case. We see that depending on the different experiments, we see that in some cases we are in the regime of efficient transduction, while in some other cases we are in the regime of inefficient transduction. And this seems to correlate very well with the kind of experiments that is performed. The scheme is the same, I only put different values, but then it appears that one experiment is always efficient transduction, the other one is always in the inefficient regime. Why so? We don't know. But this actually might be a very nice indication that since these two experiments are very different in terms of the mechanical stress that they put on the cell, it might be that this kind of two regimes emerges because of these different mechanical stresses that we put on the cell. Of course, we need much more experiments with different kind of stresses to test this hypothesis. At this level is an interesting observation we can say about the relationship between the efficiency of this transduction operation and the mechanical stress that we put on the cell to measure these flickering fluctuations. And of course, we can also estimate the kind of strategy, and we see that as more the system is becoming active, the more the strategy is information-driven. So the system tends to get more information from the internal cytoskeleton. With this, I conclude. I just leave you with the set of things that I just said, so I don't have to repeat it. These are the set of papers that more or less contain what I show, of course, much more than what I just showed here. And thanks to all my collaborators that participated in discussions or the actual papers. So thanks for your attention. Yeah.
With the active and passive cell, you think this kind of idea can be extended to other settings? For instance, this developmental processes, cell differentiation, and you can ask question of at which state of the cell differentiation the cell is more active or less active?
This specific scheme is about transduction. So as long as you can frame everything in terms of something that you don't observe, that pass information due to something that you observe, you can do it. Otherwise, probably is better to use some other scheme. If we have coupled timescales, for example, you can always estimate information. You can always estimate entropy production exactly. And you probably is better to use this specific, not this specific scheme, but something more general where you take into account all the degrees of freedom. So this scheme is very specific for the case in which you have something that you don't observe, but still contributes to the entropy production. and you want to get information on this variable without being coupled to it directly. So it's a very specific case, this scenario, this transduction problem. But in all the other cases can be extended to any system, basically. There are no particular limitations. So the framework of timescale separation is very general. You can do it you can apply to any system you want. Of course, the ability to find a solution depends a lot on the system. So it might be that you need to rely on numerical estimation or numerical sampling of the probability distribution. But in principle, yes.
No more. Left time Italian, I think.